Welcome back, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to resume the chapter the excretory products and their elimination, right? So we have already covered several topics and today we are going to see the formation of urine. Okay. So the formation of urine, right? So it is divided into several steps, right? So there are three steps of formation of urine, or we can simply say as urine formation. The first step includes the glomerular filtration, right? So in this first step, what happens is the filtration of blood by the glomerulus takes place, right? Second step is reabsorption, that the reabsorption takes place by the renal tubules, and that is the nephron, right? And the third step is the secretion, that is the tubular cell secretes what? H plus, K plus, ammonia into the filtrate, right? So these are the three main steps of the urine formation, right? So we are going to see these three of them in detail. So first, the glomerular capillaries, that is the blood pressure cause filtration of blood through three layers, that is endothelium of glomerular blood vessels, epithelium of Bowman's capsule, and the basement layer between the two membranes as ultrafiltration, right? So here what happens is when the first step takes place, right, we are going to see in that is the glomerular filtration, right? So in the glomerulus, right, the network of arteries which is present, so it is responsible for bringing the blood to the glomerulus, right? And in this glomerulus, right, the arteries opens and here the blood is being filtrated, right? So the filtration of the blood, so whatever waste is present in the blood, right, which is needed to be excluded out or eliminated out, that is all filtered, right? So all the waste is poured into the Bowman's capsule, and the glomerulus is responsible for carrying out in the filtration of the blood, right? And the filtration of the blood occurs through three layers, right? So there is presence of three layers in the glomerulus through which the filtration takes place, right? So the first layer is the endothelium of glomerular blood vessel. So whatever blood vessels which are present in the glomerulus, right, the first layer says about the endothelium, right? Second is the epithelium of the Bowman's capsule. So the first layer of the Bowman's capsule, that is epithelium of Bowman's capsule. And the third is the basement layer between two membranes, right? So the two membranes, that is endothelium and the epithelium, so there will be presence of a layer, basement layer, right, between these two membranes, right? So these are the three layers through which the glomerular filtration takes place. So glomerulus is responsible for carrying out the filtration of the blood, right, with the help of the three layers, right? So the first step says about the glomerular filtration. Now, the amount of filtrate formed by kidneys per minute is called glomerular filtration rate, and simply, in short, it is known as GFR. And for a normal person, the GFR is 125 ml per minute. So for 1 minute, or we can say in 1 minute, 125 ml of urine is being, or blood is being filtrated, right? So whatever amount of filtrate, that is filtrate, here filtrate is what? The waste which has been filtrated out from the blood. Okay. So now we are going to see about the reabsorption, right? So but before going to that, right, we are going to see that how the filtrate is formed, right? Next is glomerular filtration rate is controlled by juxtaglomerular apparatus, that is JGA. Right now this glomerular filtration rate, right, that is GFR, it is controlled with the help of the juxtaglomerular apparatus. So this apparatus is responsible for controlling the GFR of the particular percent, right? Now the 99% of filtrate has to be reabsorbed by renal tubules, which are known as reabsorption, right? Second step says about the reabsorption, that is from the filtrate, that is whatever waste has been filtered out from the blood, that is all, that is 99% out of that filtrate, it is being reabsorbed by the renal tubules, and the 1% of the waste product is being eliminated out, right? So this is saying about the second step, and that is the reabsorption by the renal tubules. And renal tubules, that means the tubular structures of the nephron, right? And the third step says about the secretion, where what happens is the tubular cells, that is the cells which is present in the tubes of the nephron, those are said to be the tubular cells, right? These tubular cells, they secrete H plus, right? Then put the hydro, like H plus ions, then K plus ions, as well as along with that ammonia, right? So they are going to secrete this into the filtrate in order to prepare the whole urine, right? So this way the urine is formed, right? So secretion, that means it is going to secrete something that is going to form the exact urine, right? So first step of the urine formation is glomerular filtration. Second is the reabsorption takes place, right? The 99% of reabsorption takes place by the renal tubules. And the third, that is the secretion by the tubular cells, and it is going to form the filtrate, right? So this is about the urine formation. Now we are going to see that what are the functions of tubules, right? So the proximal convoluted tubule, right? So that is simply known as PCT. So this proximal convoluted tubule, right? All the important nutrients, 70 to 80% electrolytes and water are reabsorbed. So the main function of PCT is to do the reabsorption of water, also along with that 70 to 80% of electrolytes, which is very needed for your body in order to maintain the constant balance of your body. And along with the water and electrolytes, several important nutrients are also being reabsorbed, right? So the first part, that is PCT, is going to do the reabsorption of nutrients, electrolytes, as well as water. Second, the loop of Henle, right? It is going to maintain the high osmolarity of medullary interstitial fluid. So this Henle's loop is going to maintain the osmolarity, that is whatever the osmosis is taking place, right? So it is going to maintain that high osmolarity of the medullary interstitial fluid. So in order the exchange which is going to take place, right, that is going, due to that exchange, the high osmolarity is maintained, and this is due to the help of the Henle's law. Third is the DCT, that is distal convoluted tubules. So this distal convoluted tubules, right, it does the function of conditional reabsorption of Na plus and water. So sodium and water molecules are being reabsorbed with the help of the DCT, that is distal convoluted tubules. Along with this, it is also maintaining the pH and sodium potassium balance. Now you know that your body needs a proper balance of the sodium and potassium, right? So this distal convoluted tubule, it is going to maintain the pH of your body, that is not only the body but the functioning of the nephron, right? So it is going to maintain the pH of the kidney, as well as also it is going to balance the sodium and potassium, right? So this is about the third, that is DCT. It is going to do the reabsorption of sodium and water molecules. Along with it, it is also responsible for maintaining the pH and the sodium potassium balance. The fourth part says about the collecting duct, where it does the functioning of the large amount of water is reabsorbed to produce the concentrated urine. Now, how the urine is produced in a concentrated manner? So, large amount or very huge amount of water is being reabsorbed by the collecting duct, and due to this, as the water content is being reabsorbed by the collecting duct, for this reason very small amount of filtrate which is formed, or we can say the urine which is formed, it becomes concentrated because more and more number of water is being reabsorbed, right? So the collecting duct, the function of collecting duct is to reabsorb large amount of water, and this is going to lead to the production of the concentrated urine, right? So this is about the functions of the tubules, right? So now the next is the mechanism of concentration urine, right? So here what happens is the flow of filtrate in two limbs of Handley's loop is in opposite direction to form counter current. Now here this mechanism of concentration of urine can also be asked as explain the counter current mechanism, right? So this is also known as counter current mechanism. And why is it so? Because the Handley's loop, right? The two limbs of Handley's loop is in opposite direction, right? And they are going to flow the filtrate in two different opposite direction. And for this reason, as the both the direction of the flow of filtrate is different, it is known as counter current mechanism. Now, the flow of blood in two limbs of vasa recta increase the osmolarity towards the inner medullary interstitium in the inner medulla. Now, what happens in this mechanism? The flow of blood, right? So whatever blood is flowed in the two limbs of vasa recta, right? So first we are going to see is about the Henle's loop, and along with the Henle's loop, side by side, we are also going to see the vasa recta, right? So in the vasa recta, the flow of blood will be going to increase the osmolarity towards the inner medullary interstitium in the inner medulla. So this we are going to see with the help of the figure. So now I'm going to show you the figure, and which is going to explain the exact mechanism of the concentration of urine. So with the help of this figure, we are going to see that how the nephron and the vasa recta is showing the counter current mechanism. So with the help of this figure, if the counter current mechanism has been asked, then you have to explain and you have to draw this figure and explain in detail about it. Now, let's start with efferent arteriole and afferent arteriole. Okay, so efferent arteriole is going to bring the blood, which is going to have the waste materials, right? And this is the glomerulus where the network of capillaries is formed, and in this is the region where the filtration of the blood is going to take place, and whatever blood has been filtered after then, that this blood is being carried with the help of the afferent arteriole, right? So afferent arteriole it is going to bring, and this is going to efferent arteriole it is going to come out from the glomerulus. Now when we are talking about the counter current mechanism. So this figure, so this tubular structure is of the vasa recta, and this is the structure of the nephron, right? So vasa recta is the artery, right? So efferent arteriole, which has been extended, or which is coming out from the glomerulus, right? It is going to come nearby to the loop of Henle in the nephron, around this nephron or the tubular part, and that is known as vasa recta. So how it is going to carry, or how it is going to do, so let's see. So now first we are going to see about the vasa recta. So as you can see over here, just have the focus on this. So first let's see about the you are going to see two things, ascending and descending, right? So you can see if it is over here, it is showing ascending, right? But when the vasa recta is showing ascending, at that time the nephrons, whatever portion or whatever filtrate is there, it is going to be in the descending manner, right? So when the ascending is observed in the vasa recta, in the nephron it will be in the descending form. Same way vice versa is for the, if the nephron is present, right? So when we are talking about the nephron, the second part, right? So here you can see this is the ascending form which is observed, but alternately or side by side, the vasa recta is going to show the descending form, right? So here, as the direction, both the direction of the flow of the filtrate is in the opposite direction, that is why this mechanism is known as counter current mechanism. So now let's see how it is taking place. So here 300 millismol, 600, 900 and 1200, right? So these are the values, right, which is showing the concentration of the urine, right? So and the three different layers is observed. This is inner medulla, this is outer medulla, and here it is cortex, right? So loop of Henle is going to be present in the inner medullary region, as we have seen in the previous lectures, right? So here we can see is that when the glomerulus, whatever the filtration is taking place, after that the filtrate is entering into the PCT, and at that time in the cortex region the concentration of the urine will be 300, right? But as it descends downwards, right, it gets concentrated and it reaches to the Henle's loop, where it is going to be present in the 1200 milliosmol, right, per liter, right? So this is what the concentration which is observed in the nephron, right, that is 1200. So here the maximum concentration of the filtrate is observed, but side by side here what happens is the water is released out, right? So when the water is released out, at that time the medium becomes concentrated. So as the water is coming out of the nephron, for this reason the filtrate becomes concentrated. But side by side what happens is the NaCl is being released out. So this NaCl enters inside this, right? And this here from, you can see over here, that is it again come back to the normal one, right? So this is what the H2O, when is released out, this becomes concentrated. And after then what happens is when the nephron is again, the loop of Henle is again, when ascending order is seen, at that time the NaCl is released out. So when the NaCl is released out, so again it becomes the like the from the concentrated part, it again becomes the diluted form. So how it is becoming the diluted form? So here when the NaCl is coming out, at that time the H2O which has came out from the vasa recta enters inside the nephron. So here, as the addition of the H2O is observed, for this reason from the concentrated form, this concentrated form is again coming back to its diluted form, right? Now, it once it reaches in the cortex region again, here the removal of the NaCl as well as the H2O takes place. And due to this, what happens is it enters inside the collecting duct. And here what happens is the removal of H2O is taking place. Not actually the removal, that means the reabsorption is been like, or the this tubular structure is going to reabsorb the water, and along with that urine is also being reabsorbed, right? So what happens is when the collecting duct is at the final, right, when it opens into the renal pelvis, what happens is whatever urine has been formed, it is in the concentrated form. So this is about the nephron and the vasa recta, which is showing the counter current mechanism, right? So this was all about the mechanism of the concentration of urine. So I hope you are very clear with today's topic. In the next class, we will see about the regulation of the kidney function, and then we will complete this chapter. So we are stopping over here our lecture, and we'll meet in the next lecture. I hope to see you again. Thank you.